Good evening. Welcome to our monthly lecture series for POOIA Professional Organization of Iranian Americans in Atlanta. Tonight, we are honored to have an eminent scientist from Georgia Tech University, namely Dr. Johannes Leisen, as our distinguished guest speaker, for which I am indebted to Dr. Bahram Hojjati, professor of Valdosta University and a good friend of Kanun and POOIA, for having introduced him to me. So many of you know what POOIA is, but for the few who do not know, POOIA is an affiliate of Persian Cultural Center, or Kanun, and is a non-religious, non-political, and purely an independent organization which predominantly invites scholars in different fields to give us a wide spectrum of information regarding their fields, in addition to human sciences and cultural issues related to our country. POOIA has a board of directors different from that of Kanun, and I have the honor of being the president of this honorable organization. All the POOIA lectures are in English so that our American and international friends, as well as our young Iranian American students or residents who unfortunately do not know Farsi, be able to participate. POOIA works tirelessly to uphold the pillars of its mission. Number one, to provide an opportunity for Iranian American professionals to network and support each other. Number two, to help the next generation of Iranian American professionals to succeed and prosper. Number three, to promote a positive image and counter the negative media depiction of Iran and Iranian people. Number four, to hold an annual gala to honor and give awards to highly achieved students from the different universities in Georgia. We just had our award ceremony on February 10, 2024, in which we gave monetary awards and certificates to 10 students from different fields. Please help POOIA for this cause by donating on POOIA.net. It is my distinct honor to present Dr. Johannes Leisen. Dr. Johannes Leisen was born and raised in Germany. He completed his research graduate studies at the Johannes Gutenberg University in Mainz, Germany, completed his research at Max Planck Institute of Polymer Research and received his PhD in 1995. After working for 2 years as a research scientist in Germany, he accepted a research position at Georgia Tech. Dr. Leisen presently serves as the researcher and director of solid-state NMR in the Georgia Tech NMR Center. Additionally, he serves as the director of the Magnetic Resonance Core Facility operated by the Institute of Bioscience and Biotechnology and the Department of Biomedical Engineering at Georgia Tech. Dr. Leisen has mentored and trained hundreds of students and postdoctoral students in NMR and MRI experiments and published more than 100 scientific papers in various journals. Tonight, Dr. Leisen will be talking about MRI, its fundamentals and applications beyond medicine. On behalf of POOIA, Professional Organization of Iranian Americans, and Persian Cultural Center, Kanun, I express my heartfelt appreciation to you for having accepted my invitation to talk for us. It is indeed an honor for us to have you as a distinguished scholar presenting for our participants of POOIA. Thank you so much, Dr. Leisen. We are all ears. Would you please start?
Thank you very much. Let me see, but I get started with sharing the screen. So thank you very much for this kind invitation, and thank you to Barry to sort of rekindle an old friendship. It was really good to hear back from an old friend, and I'm really really delighted for this invitation. Also, thank you, by the way, for my wife who is in the audience, and yeah. So I'm research scientist at Georgia Tech. Here's the biotech quadrangle where I work, and take this as an invitation to visit Georgia Tech. We have open houses. We have all kinds of opportunities to come, especially send your children and maybe also your grandchildren to Georgia Tech. We are an excellent school and we offer excellent programs, and we have a lot of international students. So we have a really fruitful research environment and teaching environment also. In my position, just especially since Dr. Garmenstani is listening, I'm in charge of 2 laboratories. Both laboratories are walk-in facilities, meaning it's not our philosophy that we have technicians do experiment for people. Our Georgia Tech philosophy is that we train people to do the experiments. So the idea is I like to call myself as someone who gives paper-to-paper support. What I mean by that, someone comes as a student, a postdoc, graduate, undergraduate student comes with a research paper and say, hey, I want to do something rather comparable to that one. I have maybe synthesized something, I have a new system. I will look through this paper, we discuss this paper, we discuss new experiments, we set these experiments up, we make these experiments work. I train this person to do the experiments. Then I let sort of hold hands, let hands go. This person does the experiments. And eventually the person has data. I help with the data analysis. I help writing the paper. So that's how I like to operate and how I like to think I operate. So I'm in charge of this laboratory, and here's the website of the other laboratory, which is an MRI instrument. Okay, I'm trying to do a tightrope walk now. In a way, I tried not to be boring and trivial for the experts. On the other way, I try to maybe make it interesting for the non-experts. So it might be too difficult for one, too boring to the others. I try to foster to everyone. If I don't manage, if it's too trivial, my apology. If it's too difficult or gets boring, my apology as well. I try the tight walk. There's 2 parts. I try to explain at least a little bit how MRI is even working and what's behind of that. And then I will be what's called sound bites, very quick snippets about probably interesting, unusual applications people might not have thought about. Okay. Let's start with the beginning. And I was asked about this gentleman before already. Max Planck is a very famous physicist. He's called the father of quantum mechanics. Now, what is quantum mechanics? Quantum mechanics is the physics of very small objects. Interesting enough, Philippe von Joly was the physics teacher of Max Planck, and Max Planck, being the very famous, very achieved physicist as a young man, he was advised to stay away from physics. Physics is a highly developed, nearly fully matured science that, through the crowning achievement of discovery of the principle of conservation of energy, will arguably soon take its final stable form. So with advice, there's nothing to find anymore. We are soon finished with physics. We know everything. This was in 1878. By now, we still don't know everything. So this was clearly a wrong advice. The advice was also, why don't you turn to economy? Now, I think Max Planck turned a little bit to economy because he discovered something in economy, which I, not being economist, sort of thought I recently discovered as well, which then translates to his fundamental finding of physics. Economy is also works in quantum. If I have $100 and I want to divide $100 by 3, I cannot do that. I will have $30, 3 times $30, 3 times $3, and then 3 times 30 cents, 3 times 1 cent. But okay, there's something missing. What about this 1 cent which is left over? I cannot count. So there is a quantum in business also, which is given by our currency. I cannot divide our currency as much I want. The same thing is Max Planck's discovery hypothesis, proven hypothesis, that the energy of small objects does not come in individual, in absolute small numbers as well. It comes in quantums. So an energy can be only so and so, have only a discrete value, can have so and such of a value. This has really dramatic consequences. The math is going to be incredible, complicated. It's not the simple math anymore. Here's the two main principles of quantum mechanics, or the two axioms of quantum. There's the uncertainty principle and Schrodinger's equation. Here's the mathematical description. If you remember your basic math from elementary school, there's the law of commutation. X times p minus p times x is 0. No, not in quantum mechanics. And this means this cannot be just numbers. This must be something else. And Schrodinger's equation, the other fundamental equation of quantum mechanics, is even stranger. The principle of the Schrödinger equation is that you actually cannot observe things. The essence of something is here the psi, which is the wave function. But any object which only becomes observable, measurable, by having sort of an actuator on this wave function, which gives the observable, which is the energy. We have here this young lady who is clearly observable by this gentleman if he looks at her. However, if he looks away, if he's not observing it, she's just a wave function, an superposition of various states, which is clearly not happening. He says this is not happening in big objects, but in very small objects, it's different. Keep in mind, if something gets very small, to see it, you need to obviously shine light on it. If the light has associated with energy, and you have a very small object, the energy of this light can change already the small object. That's more or less what's behind this equation here. Okay, now, based on these equations, we can make big strides. For instance, we can understand atoms. Now, this is the kids page from a long time ago in the Atlanta Journal. And I find this, so even kids know that we have odd atoms. Just, this is what we learn in school. An atom consists of two major parts, the nucleus. And around this nucleus is positively charged. You have electrons, which are kind of moving in defined space around the nucleus. That's the core of everything. That's the atom. And we can very well understand, based on quantum mechanics, we can understand why there is different elements and how chemical reactions and why they react and what the property. A lot of things. What is concerned with atoms and molecules, we really can only understand with quantum mechanics. Now, a very important experiment for quantum mechanics was the Stern, I pronounce it German, the Stern-Gerlach experiment. Gerlach is his name. He was the experiment, he was the theoretician. So what did they do? They had an oven. In this very hot oven, they had silver, such hot that at the outlet of this oven, silver atoms would escape. And it went to kind of two pinhole cameras to have kind of a beam of silver atoms, which then went through a magnet. And the expectation was that one just sees a line which kind of reflects the thermal energy. But the observation, the very unexpected observation, that there was just two dots instead one line. The two dots can only be explained with quantum mechanics. It's explained with a specific quantum mechanic property, which is called the spin. While this goes to the magnet, the electron associated in this sort of the one free electron in the silver sees the spin, which causes different energy levels. So there's two energy levels for the spin. We just have two dots. Now the spin, we could think of it of a rotating charged particle in the magnetic field, but this is not the full representation. A much better representation is this here, this sort of vector moving, and okay, we don't go too far in it, but this is a much better representation of what's happening than that one. And even this is not very good representation. A much better representation is that one. We don't go into details now. This is the quantum mechanical description of the spin. We start with this one, Schrödinger's equation, and we solve it. And so, okay, good. So we don't go into this one, but the important part is spin is a quantum system. There's two energy levels. The spin system, meaning the electron in the magnetic field, can have an up state and a down state, and cannot be anything in between. There's a big difference between two big objects. We can take, assumingly, all kinds of energy levels. The spin of an electron can only have two. Now, this was shown in 1922. In 1946, so now physics moves to the United States. Those two gentlemen, Felix Bloch and Edward Mills Purcell, did an experiment which gave them the Nobel Prize. They actually found that not only the electron here, but also the nucleus has a spin. And they had a much significantly more complicated setup. And this is actually what's in this original publication, which is a Nobel Prize winning publication. Doesn't seek too much as to get a Nobel Prize if you look at these curves here. But that was it. Here's the setup of their experiment and from the original publication. But to explain a little bit more what they were doing, they had here the sample which contains the spin. It was just water, and this water contained the nuclei of the hydrogen atoms. Water is H2O, the hydrogen and the oxygen. So the hydrogens of this water were put into the magnetic field, and they were sending radio frequency to this field. Now the radio frequencies, if they come with the right frequencies, they sort of tickle those nuclei, and they will respond. They respond by taking up the energy, and they respond by sending energy back. So we send radio frequencies on our sample, the sample reacts, and it sends radio frequency back. We have a radio sender and a radio receiver. And we come up again, math. Sometimes stuff is easier to explain if you know a little bit of math, and it's easier. The frequency here of the radio frequency is proportional. It depends on this magnetic field. If the magnetic field gets higher, obviously the frequency gets higher as well. And the higher the frequency, the more sensitive the experiment is also. Therefore, one wants to have really, really high magnetic fields. Now, here's the first experiment. One thing that physicists very quickly discovered was also, we have the magnetic field, we have something else. This is just a constant, but we have the sigma here. And the sigma is called the chemical shift. The chemical shift actually is a probe of the environment, because it turns out that this frequency not only depends on the magnetic field, it also depends on the chemical environment. Now let's look at the first example of the first measured real NMR spectrum. This here is ethanol, simple alcohol. Alcohol has two carbons, which one contains three hydrogens, one contains two hydrogens. This bond to an oxygen, which is also a hydrogen. We have three different types of hydrogen, depending where it binds. 1, 2, 3 peaks, like signals here. And if you look, we have 1, 2, 3. If you look at the intensities, or better, the integral of those peaks, the intensity is also 1 to 2 to 3. So I have here a very, very great tool to actually probe my chemical structure. From this here, I can see I have three different environments for my carbon. I have a tool to actually probe my chemical structure, which is very unique, very great, because that's what chemists just have been dreaming about. Physicists were happy. Okay, now we have the chemical shift, but chemists, and here's the leading head of the chemists, Richard Ernst, chemists were really excited. We have now a tool where we can characterize chemical structures of molecules. So Richard Ernst, also Nobel Prize winner for chemistry now. was really fundamental for developing NMR as a technique. He went to making it multidimensional, showing spectra. He was also one of the major heads of MRI. However, his interest as a chemist was not in MRI. There's some fundamental papers. The Nobel Prize was mentioning him as one of the leaders of MRI, but that's where he contributed, but he sort of didn't follow up in this field. Here he is his autobiography, working as a young man in one of the very early NMR instruments. That's how these instruments have evolved now. That is not even the latest. Here's the UFO instrument, 900 megahertz. And the spectra are not simple lines with 3 peaks. You can go 2-dimensional now, where you correlate different nuclei which have spins, and one kind of get 3-dimensional maps, and one can go up to measuring very, very complex molecules such as proteins, DNA, and so on. So this is by now a very evolved technique, and we follow this and use this at Georgia Tech also in our NMR laboratory. So it's part 1 of my job. Now part 2 is, and here comes our friend, we mentioned him also already in our chat before this talk, Paul Lauterbur. Paul Lauterbur was the winner for the Nobel Prize in Medicine together with Peter Mansfield. And he was sort of the head of MRI. The Nobel Prize in Medicine for MRI has an interesting story, because there was also, I should say, the fairy who was not invited to the wedding, which was Raymond Damadian. Damadian probably was not an academic researcher, and he did the following. He probably made some invention with regard to MRI, which had, according to experts, probably some flaws, but he patented them. And for patents, you do not get the Nobel Prize. He had the company still exists. He had a successful company, but when this Nobel Prize was awarded, Damadian and his company sort of put out whole page advertisements or paid pages in the New York Times and other reputable journals, claiming that he was left out in the Nobel Prize. Anyhow, going back, here's the first MR image of an human, and this is actually the human wrist. This is actually very interesting. So here's this team who sort of developed this, had the first really real sample. Really interesting. They display MRI images different than they are currently. I think I have this in the next slide. Current MRI images would, according to current MRI standard, would according to that, areas with high tissue content, high more amount of, will be kind of bright. They displayed it differently. Black, dark areas indicate regions that contain high concentration of mobile protons. Sorry. Today, it's usually done differently. Why did they do this? I have a theory here. They are very well aware of the impact this image had, and they reminded of that one here. This is the first X-ray taken in 1895 by this gentleman, Wilhelm Röntgen, the inventor of X-ray, or discoverer of X-rays. This is actually not his image, but this is the image of his wife. He seemed to be a little bit of a show-off, because this is probably the Christmas gift to his wife, the big ring she's wearing here. But anyhow, maybe he thought, or he gave his wife this honor to have this first X-ray published with not with his hand, but the hand of his wife. Okay, now what is the MRI? How do one actually get the images? So what did Lauterbur and Ernst realize what needs to be done to get an image? We have this equation here. Omega, this is the frequency again, depends on the magnetic field. Now, if the magnetic field over our object is not the same everywhere, but if different areas in the object see a different magnetic field, this will be reflected also here in the frequency X. So if I send out here and I have another magnetic field, I have the big magnet, a small magnet, which creates different magnetic field around this object, I will see a response which kind of reflects my object. A little bit reflected here. So we have here two objects. If those two objects sit in an homogeneous magnetic field, meaning every area of this object sees the same magnetic field, we just see one line. There's no spatial distinction. If every area in this object sees a different magnetic field, we see this distinction. And if we have a different magnetic field just going in one direction here, if this is a linear variation, we see kind of here the profile. Here's the kind of the cylinder, and here's kind of the square here. You see this reflection here. And Lauterbur was doing something. He just rotated the gradients, and from different gradient orient you get different images. And those images allowed him to reconstruct, and to my knowledge, this was a very simple reconstruction with just a ruler or so to come up with this first MR image. So that's the setup of an MR scanner. We have a big magnet on the outside, and the person comes into this magnet, and we have radio frequencies we send, and we have gradient coils. Those are the switch magnets which cause the images. The big difference, and why is MRI interesting in comparison to X-ray? They pretty much do the same. The one thing is X-ray is very good to measure hard tissue, bones. MRI is just the opposite. It measures the soft parts, the water, the free hydrogens. So MRI is very good to measure the soft tissue. It comes with the added advantage. X-ray has ionizing radiation, meaning radiation which is able to break chemical bonds. If it can break a chemical bond, the chemical bond can go somewhere and can cause damage. It can go to the DNA, which might cause cancer. If it's non-ionizing, it can at best heat up the body a little bit. So this is a much more patient-friendly technique than the X-ray. Obviously, it's also more expensive. Okay, so I'm not talking about MRI in the hospital. I just say MRI in a hospital is a technique which is highly developed for one type of sample, which is the human patient. Now, if not used for people, MRI can also use for animals. It is an expensive technique, but there is very expensive animals also here. I'm sure this is a very nice horse, and it takes a lot of effort to put a horse into an MRI scanner like this. But if a horse has injury and it's a racehorse, also a lot of money can be involved in the proper healing and diagnosis of anything with the joints and the bones of a horse. So much that by now you can even buy a dedicated MRI system just for measuring joints of horses. Here, this is the company. Of course, a system like this where the horse can just walk in and you put the leg here is much more horse-friendly than a system like that. And people love their animals. By now, there's also in Atlanta, at least one I'm aware of, veterinary clinics which offer MRIs of your dogs and your cats. There's people willing to pay a lot of money to have their dog or their cat live a few years longer, and people send cats for chemotherapies which require MRI supervision or so. Those things exist. However, my hypothesis, I'm pretty sure that most of the MRI of animals is done what's called preclinical research, and preclinical research is for the most part the research done on animals, and for the big part mice and rats. It's research which is done to develop medical techniques before they're actually going to be used on humans. So here's our MRI scanner, which is used a lot for animal studies. Here's one of the nicest brain tumors, images which we ever measured. And you see really nicely the tumor growing in the brain of a hat. This is a good deal which is done with this particular instrument. However, this instrument also is done for for research which is really not related to animals. But to come up with one idea about an animal study which is currently ongoing is the research of Dr. Kostas Avanitis at Georgia Tech. He is studying tumor growth and tumor therapies using focused ultrasound, FUS, in combination with MRI. So what happens? We have the mouse, and the mouse is this is actually a special mouse. The mouse is injected with tumor cells. So these tumor cells grow, and you need to watch the growth of the tumor cells. Guess how it's done? MRI. Before even admitting a mouse to the study, we want to now to study tumor growth, to study any type of tumor medication. We want to see whether these animals even, whether tumor is even there. So you can take an MRI. You take MRI, and after 2 weeks or so, tumor should have been grown. The animal then goes to the MRI for FUS setup. Within the MRI, we apply ultrasound, and we also measure the MRI here with this. This is the simple, all this thing is in the magnet. This way, we can apply the ultrasound, and we can use the MRI through a special technique to actually measure the heating. So as such, we kind of measure the temperature in the brain, and we feed the temperature, which we measure back to our ultrasound, that we actually can regulate the brain, the temperature of a specific area in the brain. And unfortunately, this doesn't end well for the rat with the tumor. Even though it might heal the tumor, the rat will kind of be euthanized, and it will cut open. Histology will be done to see what actually happens with the brain. Here's again a little bit. Here's this area of the brain which is heated up. This area stays colder, and we can sort of measure. Here's the temperature. We use this image to control, use the feedback loop to control the temperature. So we can selectively heat our brain temperature for this area from body temperature to here 40 centigrade. And then when we switch off our ultrasound, it goes down again. Now the experiment here, and here you see one of those laboratory mice. Laboratory mice. This mouse probably, if I would release it, it wouldn't live too long in the outside, because this is an immunodeficient mouse. I can inject tumor. It doesn't have any immune system. Therefore, the tumor would grow. Ideal system for tumor studies. Those animals live under very clean, sterile conditions. Here we injected the tumor in the hind leg, and you see it growing. This was a study where contrast agents were developed. An agent which kind of is a special microparticle, nanoparticle, which contains something which binds to the tumor. It's injected, it finds its way to the tumor where it binds. We see it with the MRI, and it even has a potential to kind of this nanoparticles, they absorbed again radio frequencies and heat. So this is a finding, detecting, treating with just one particle study. Okay, another interesting study with animals. So this is the group of Dr. David Hu at Georgia Tech. David Hu is a biophysicist. He looks at all kinds of odd and interesting things. He wrote a book about this even. He looks at one of his interests is snakes. So how does a snake crawl? And there's different type of motion a type of snake can have. This is what's called the concertina locomotion. If the snake is in a very tight space, it kind of forms these type of waves, and then it straightens again. That's the way how it moves forward. Now, to make a real biophysics, you want to know how much energy actually is the snake using up for this type of motion, and how can I measure the energy used for this type of motion? Now, how can you ever measure the energy used up by a snake? You could do this by making the whole exercise in a container which measures the energy or the heat sent out by the snake, which really does not work. So the trick, and which we borrowed from some other studies on humans, is the following. What actually measures energy? The energy in the body is determined by the ATP. ATP, adenosine triphosphate, is a molecule which stores the energy, and we can measure now the spectrum. This is not hydrogen, this is phosphorus. And we kind of see our ATP here, and this kind of reacts to inorganic phosphorus, which we say here. We can take the ratio. So from the ATP concentration, we can more or less figure out how much the snake has been consumed. And we can, if we wait with us for a couple of minutes, we see how the body sort of reproduces the ATP for as a function of the resting. So the snake here, we had sort of kind of a ramp as to what pieces the snake was put in. We had the snake crawl up here, crawl up this ramp and put them down. And the graduate student loved that, working with the snake. The snake would be crawl, do it several times until the snake is tired and snake just wouldn't crawl anymore. Interesting, the way to attract snake-like dark. So just a piece of paper blackened with a Sharpie made the snake crawl up there all the time until it was tired. When it was tired, it went into this kind of, we had several iterations at the end. We had sort of kind of this plastic tube which had holes for the snake to breathe. It went right away into the MRI scanner, and we took, here's an image, and we measured this phosphorus spectrum. Took a lot of efforts. Like one idea we had, we anesthetized the snake. How do you anesthetize this animals? You give them an anesthesia gas. The rat breathes the anesthesia gas, 1 minute it sleeps. When you stop the anesthesia gas, 1 minute it wakes up. The snake, you give the snake the anesthesia gas. The snake can close its nostrils and decide, I'm not breathing anymore. Half an hour later, the snake decides, okay, I can open my nostrils now. And then it falls asleep. But half an hour, it will have recovered here already. So this didn't work. However, snakes are cold-blooded animals. They don't move too much anymore if they get cold. And it's a little bit cold in the MRI scanner, a little bit cold in this hole here. So we just put it here, and it barely wouldn't move. We had very little images, experiments which did not work, where the snake was not moving. The very unfortunate story of this one now, we only had 2 snakes to do this experiment. Then we had a nice paper written, send it out to a journal, and the journal came back, no, this is a biological study, and biological data are only relevant if we have at least 20 different object studies. In the way they write, it was kind of a pilot study, but we had sort of reproducible results for 2 snakes. This was the end of it. Now I'm happy I can at least present it once or so. Now, okay, going beyond, we leave animals now. Food. I think a lot of people had a lot of fun with the MRI scanner by putting pineapples. This is a pomegranate, this is a tomato, and this is a cucumber. We had it just 4 weeks ago or so, 8 weeks ago. Georgia Tech had a science and engineering day. People came and we had a lot of fun. We put food in the MRI scanner, and we had kids coming to the MRI lab, and they could guess what food it is. And but there's more to it. If you look at the publications about MRI and food stuff, you find a really high proportion of publications coming from Japan. And Japan, I haven't found this anywhere published, but a very knowledgeable person in MRI told this to me. Well, what's in Japan, people are willing to pay a lot for fruits. Like people are willing to pay $1,000 for cantaloupe or so. And this is really highly luxury good. And people are willing to, if you pay $1,000 for cantaloupe, it needs to be perfect. One way to check that the cantaloupe is perfect is to have an MRI scan before. So there is some applications for high luxury fruits to actually do quality control on those. Otherwise, it's probably too expensive. There is research probably in food science. Here's, for instance, bread baking. If you have an industrial oven or so, an industrial baking process, and you want to look at temperature distribution, baking processes, and so on, you can do this, for instance, either by constructing a heating device within the MRI scanner where you observe the baking in situ or taking baked products out. And we had several food stuff in this sense measured over the years also. Here, my friend Barry, it's long time ago, but this is actually stuff which Barry and I did years ago back here. What we have done at Georgia Tech is we looked at textiles, also known as engineered fibrous substrate. We can look at woven fabrics, get the woven structure. We can look at non-woven felts, diapers, carpets. We looked at the drying process in carpets, see what happens if you force air to the carpet and see how the carpet, let the carpet dry. We looked at papers. Here's the moisture ingress in a very thick piece of paper, and we see how moisture would be increasing. And this we modeled this. This data was Barry and I produced this together. Okay, another application. Cultural heritage. We're talking about mummies. We're talking about frescoes, famous paintings, stuff which is in a cave, in a church, at places where it probably cannot be removed. A fresco in a church, which is maybe decaying because there's water behind the fresco. This can be characterized also by MRI techniques, NMR techniques. The problem is you cannot take the fresco, cannot take this out of the museum, out of the church, out of the wherever it is. So there are special developments of surface MRI devices. So here's this device which is brought close to the fresco, and it actually can measure up to a depth of 1 centimeter what is actually within this surface on the within this picture. And there's several devices. There's an end industry. You can buy a device like that, a mobile surface explorer, which is MRI based. Next application. That's actually data from a successor of my previous job in Germany. He was the first one, to my knowledge, who looked at fossils. Now, the story goes he was in an open house or talk, public presentation. He also explained MRI and said, same as I, well, this works very well for soft tissue, for hard tissue, it doesn't work. And then someone came, oh, can you please measure my fossils? And the reaction was, didn't you just listen? Fossils are hard, obviously, there's a stone. You will not see anything, but okay, since it's you, I will try it. And actually he took images of the fossils, and he could, this is kind of like an ancient, I don't know what, snail. And actually, to his surprise, you can take images. So this is probably not so much interesting for the fossil, but for the geologist or the mineralogist, it is very interesting because fossils of the water which is trapped in the fossils, this is water which is close to impossible to remove. It is sort of bound into the bound by the geological or mineralogical structure, but it is mobile water which allows the recording of structures of fossils. Of course, fossils also very valuable pieces which probably cannot be simply cut open. Okay, talking about engineering, big interest in engineering. One of the really powerful things MRI can do, it can measure not only pictures, it can also measure flow. So here's an example of a research group in Great Britain. They had sort of a bed with beads, and they can measure the velocity profile of water which is flowing through this to the sample. We did this as well. We were interested in flow through a textile sample. So we have a setup here where we have kind of a tube which goes all the way through our fancy magnet, and our sample sits here at the center. And we need to make great pride to make sure that we don't have any air bubbles in there, because this is really and with that, we can also measure velocity profiles. Here's our experiment, and here's our simulation. The simulation, one can simulate these things very well, but the simulations are only as good as you can sort of verify it with an experiment, which we sort of could do for rather complex piece of fabric. Can also do this. This is another data produced at Georgia Tech. Here's an artificial heart valve. Not in the heart, but in the flow setup. And we can characterize the flow through this artificial heart valve. This technique now from engineering can also be used in clinical MRI. And we are actually in the process of a study, conducting a study where we also use it in mice, where we measure actually velocities of blood flow into the brain as a function of the heart cycle, which is quite a challenge, but we seem to have data. Okay, coming close to an end, we have seen that we can detect chemical structures with NMR. Here's something, just a little expansion. So far we talked about the hydrogen. We can use the phosphorus. One can also use the carbon. Here's our NMR spectrometer R2D2. Unfortunately, R2D2 is a 1985 design also, which broke just a few months ago. And you collect money to buy a new one, which is very expensive. But here's our R2D2 magnets, which we used. Okay, now, so we can use this to measure kind of structure, which tells you the chemical structure. You can combine this with the image. So what you can do, you can measure an image of a rat here. You select a particular area here in your images, and for this area, you measure kind of the hydrogen structure. So for particular areas in your brain, you can get the chemical structure. This is very powerful. If you do, let's say, a tumor study, we can measure the size of the tumor, we can characterize the tumor, but if you want to see if a like a medical treatment does something, it takes like 2 weeks or so to see if the tumor shrinks, if something happens. It takes less than an hour to see if a medication is doing something, like changing the biochemistry. So we have here a tool to more or less selectively measure biochemistry as function of the location in the brain. Finally, last not least, the most amazing thing what MRI can do, detecting thoughts. It's not as great as here, but there is functional MRI or fMRI, the technique which allows you to look into your brain and actually allows you to see what your brain is actually doing. So how does this work? We have the brain, and depending if we are thinking about something, depending what we think, certain areas of the brain get active, and the oxygen content in these areas will change. So for certain things, if I do, for instance, math, other areas of the brain will be used than if I'm just coming up with something, if I read a book or so. These areas which will active will have the increased oxygen content, which will have a slight effect in the MR image. This is sort of the relaxation, the response to our radio frequency pulses will change. This is an effect by 2% or so, but we can see it. In a way, we have a way now to unlock the mystery of the mind. And this is what's functional MRI. Here's just one example a colleague gave. We asked someone who knows sign language. We actually tie these hands of this person, put this person in an MRI scanner, but say now think chair, think this hand movement, and you'll see certain areas in the brain gets activated in cells. Now think you're going to lie down on your bed now. Other areas are going to be activated, and think now it's going to get hot again. Other areas are being activated. Now this sounds like a great technique. The caveat is what you see here is not the experiment of one person. The effect is tiny. This is an effect of maybe 20 persons. 20 persons do this experiment, and it is kind of really convoluted area. That this is the average of 20 persons, and this is not the brain here is not being measured. The only thing is those dots are going to be measured. The brain, this is a separate standard brain onto which the data measured are going to project it. So it's really not the technique yet where you put a person in and you can figure out what this person is thinking. But you can certainly get insight in the function of the brain. Many studies, for instance, also wouldn't take me since I'm left-handed. They exclude left-handed persons. This is supposed to be totally different. And they like to take only strictly right-handed persons for this technique. Still, multi-million-dollar instruments. Georgia Tech has another person, has one human MRI scanner at CABI. That's the Center for Advanced Brain Imaging. That's where these images are from. CABI is run by the Department of Psychology together with Georgia State and solely for these type of studies. With that, I have given sufficient information. I like to put an acknowledgement out, which is based on a Talmudic wisdom. I would like to thank all my teachers from whom I learned a lot, all my colleagues from whom I learned more, but most of all, I like to thank my students, with whom I work on a day-to-day basis, from whom I learned most. And I will thank you. I'm sure your brain is buzzing now. This is the mascot of Georgia Tech. I like to thank you for your attention. Thank you very much.